Aloha and thank you for coming. Today I'll be talking a bit about a case study of teaching scientific literacy in a remotely taught animal behavior lab. The COVID-19 pandemic forced many of us into teaching and learning situations that were far from our ideal, including many remote learning opportunities and modalities. At BYU Hawaii, we were entirely remote and asynchronous for fall of 2020. And during that semester, I taught a remote animal behavior course and lab. Now, a major goal of my lab course is typically to teach the process of conducting animal behavior research, which we normally do by doing a semester-long research project. And this was not feasible given that we were off campus and spread across 6 different time zones. So I looked for an alternative. One silver lining, I think, of the pandemic was that it brought out a great collaborative spirit amongst educators in many different fields, including animal behavior. I was thrilled to find and benefit from this animal behavior and evolution teaching exchange Facebook group, which actually worked out so well that some ambitious members ended up publishing this great primer for teaching animal behavior online, which I encourage you all to check out. So one approach that I encountered through that group was the C.R.E.A.T.E method for teaching the process of science and improving scientific literacy. So after looking into this method, I decided to structure my animal behavior lab around this approach. If you're not familiar, this is an idea that was developed by Sally Hoskins and others. And the general approach is that students are guided through a deep dive into a specific series of research articles, completing a number of different activities that give students greater insight into the scientific process. And this method was particularly appealing to me because BYU Hawaii has a very diverse student body. And research by Leslie Stevens and Sally Hoskins has shown that this method produced multiple gains in diverse students. So the C.R.E.A.T.E acronym stands for Consider, Read, Elucidate the Hypothesis, Analyze and Interpret the Data, and Think of the Next Experiment. And for this work, one must select sequential scientific studies to work through in groups and as a class. And I decided to work through 3 articles by Dr. Corinne Finnig at UNC Chapel Hill and co-authors that built on one another in a sequential way. And these papers dealt with hybridization behavior and spadefoot toads and were a great fit for this exercise. So for the rest of the talk, I will walk you through the process that we followed and provide some insights from my experience doing this remotely. The way I structured the course was that we would spend 4 weeks on each scientific paper. And this is where you could kind of parse this out and just do one paper if you would like to spend a shorter amount of time on this method within a course. In my case, I put students into groups of 4, and I tried to sort of group them by time zone if possible so they could coordinate their meetings. And I gave them clean versions of the publications. That is, they would only get the text and the figures with no title or author information, and they would get them section by section. And the process I'm going to describe to you is repeated for all of the papers that we read, all 3 of those papers. So for week 1, students would consider. Here they would create concept maps outlining major themes from the introduction. They would read the methods and results, and they would diagram experiments through cartoons that would kind of demonstrate what they actually did in the experiment. And they would also annotate figures. And then at the end of each week, I would have individual meetings with these groups. And this was a great opportunity to kind of debrief, answer questions about the process, especially that first time through where concept mapping and these other approaches were kind of new to them, and answer any questions that they had and sort of provide some feedback along the way. In week 2, they would elucidate the hypothesis. Here they go through and they clearly define the hypotheses being tested by each experiment. They then analyze and interpret the results. Here they summarize the results of the study through concept maps and bulleted lists. And then they have a group meeting with me again. Week 3, they think of the next experiment. So here it gets really exciting. They designed 2 suitable follow-up experiments that would build on the focal study that we've been reading. And I found this particular part really exciting for students because they would in many cases go completely different directions from the actual follow-up study. In some cases, they would propose something that was almost identical to the follow-up study. But here I think there's a lot of opportunity for students to get creative and connect to different topics that they're learning about in the course. I'd also meet with them here, and then during that final week, we would have a grant panel. And so what we would do here is that each group of 4 would receive and review 4 proposals from other groups. They would rank them, and they would, based on different criteria that I would outline, and then we would have a class Zoom discussion about the nature of the scientific process, about scientific progress, about granting, about how one goes from one study to the other. And this was a really interesting part for the students to be able to see kind of how that process unfolds from the perspective of a researcher. So in the next few slides, I'm just going to show you a few examples of student work. I'm not going to spend much time on these, but feel free to pause and check them out later. Here are 2 examples of concept maps that students made from introductions. Here are some annotated figures and tables, along with a cartoon representation of one of the experiments. Here's a student's work detailing the hypotheses that were being tested by each experiment. Here's a clip from an analysis template that students use to dissect tables and figures. And then here are a couple figures that I took from next experiment proposals that I found pretty creative. So after completing this process for all 3 papers, students generated questions about the process to ask one of the authors of the studies. And then we had this culminating experience, which was to meet for about an hour over Zoom with Corinne Finnig, who was a PI over this work. This allowed students to get a glimpse, even more so, behind the curtain and ask about why they did certain things, how she ended up asking the questions that she did and designing the experiments that she did. And I'm just going to provide a couple examples of student feedback that I think are really insightful. So this student says, one thing I really liked was when they ended up getting their studies on hybridization kind of by accident. So if I were to pick up something from her, it would be to make sure to pay attention to other unexpected data during research, because that could lead to a new and interesting discovery. And the second one, hearing all that made science work and scientists seem more imperfect and relatable, honestly. Now I feel that I'm not too far away, maybe, from being a scientist myself. If I can just have her drive and work ethic, along with the professionalism and problem solving to get over the funding and mistake roadblocks, I can do it too. Nothing that she said sounded too unrealistic or daunting, just lots of hard work. So I found this process incredibly helpful in the virtual modality, and I also think it works great in person. Feel free to email me if you've got questions. I'm happy to provide instructions and syllabi from my course. Thank you all for listening. Thank you to BYU Hawaii for their support, my students, and also the Behavior and Evolution Teaching Exchange Facebook group. Mahalo.